This week we're going to be talking about the nervous system. I'm going to try something a little bit new this week. I noticed these videos not only take a really long time to export and upload, slowing down the quickness that I'm able to roll them out to you all, but also it's kind of a lot to sit through an hour long or 45-minute long video. So I'm going to start trying to break these lectures into two parts, aiming for about 15 to 20-minute videos. So maybe that is a little more digestible for you all and a little less laggy as far as buffering and all that kind of stuff. So anyway, we will be talking about the nervous system. The nervous system is absolutely one of my favorite systems to go over in anatomy and physiology. I think it is incredibly interesting. I hope that you do as well. As always, this system is fun, but it can be a little complicated because we deal with some concepts that we haven't really touched on before. So if you're having a hard time grabbing the content, it's just not sticking, whatever, please reach out to me. I'm always happy to meet up or email or text or whatever to get you all to figure out this content. That is one of my favorite parts of my whole job. So please, please, please don't struggle. Don't feel like you're bothering me. You're never bothering me. Okay, so diving in, the nervous system. Our nervous system is going to have a few different functions, but it's all going to kind of coordinate into the same overarching theme. And that is going to be to control, coordinate, and communicate all of the functions of the whole body. So this is our control center of the whole body. All right, we're going to have a few different ways that this works. We're going to kind of have a pathway that this information flows through, and that's what I'm going to talk you through right now. The first step in this system is we're going to have sensory input. We also call this afferent input. This is where we are gathering information from our environment. So these examples here, you're seeing something, right? In this, it's a glass of water, but you're seeing something in your environment. Your eyes are taking in that information. You're sending that to your brain. Step two is integration. This is, at this point, you're in the brain is processing this information. The brain is making a decision based on this information, and then it is sending that decision out to the motor output. We also call this efferent. An easy way to remember this is your efferent output is exiting the brain. Okay, so the brain is sending this to the motor system, and it is providing this response to the information it's giving. So like in this example, an arm is reaching out to grab this glass of water and say, hey, I'm thirsty. So that is kind of how this information pathway works in the nervous system. Here's just another look at the same pathway that I included, just so you all have multiple study tools while you're working through this. So you can see here, there's a pin pricking the skin. The skin is taking in that sensory input, right? So we're taking that afferent input. It's going into the spinal cord because it's following all of those nerves all the way down into the spinal cord. The spinal cord is sending it to the brain. In the brain, we're processing that information, and then we're sending out motor information to tell our skin to, hey, pull back away from that stimulus. So that's just, I'm just trying to give you guys a few different ways to conceptualize that same thing. So that's why sometimes I throw in a few different pictures of kind of the same thing, illustrated a few different ways. So the structure of the nervous system, we're going to have a central nervous system. We refer to this also as the CNS. This is going to consist of your brain and your spinal cord. And we're also going to have a peripheral nervous system. We call this the PNS. These are going to be the nerves outside the brain and the spinal cord. So we're going to have 31 pairs of spinal nerves, 12 pairs of cranial nerves, and all of the roots and branching nerves that go throughout the body. For our nervous tissue, we're going to have neurons. Neurons are what we call our nerve cells. The purpose of our neurons is to transmit messages to communicate with other cells. We are going to have a few different cell types of like helper cells. We refer to these as neuroglial cells. You may also just hear these referred to as glial cells. Sometimes people drop the neuro because we are talking about the nervous system, so almost everything is going to have a neuro prefix in front of it. So our glial cells are going to be cells that support, insulate, and protect the neurons. So they, again, are helper cells. We're going to talk about a few different types of those. We have astrocytes. Astrocytes, their main purpose is to supply nutrients. We also have microglia. These are going to help with immune support. We have ependymal cells. These are going to make our cerebrospinal fluid, and we'll talk more about the purpose of cerebrospinal fluid later. We will have oligodendrocytes. These are what form our myelin sheaths in the central nervous systems. We have satellite cells. These are going to support the neurons in the peripheral nervous system. And we also have Schwann cells. You will also, so Schwann cells is the old name for this. You will also hear these referred to as neurolimocytes. And the purpose of the Schwann cells is to support the neurons in the peripheral nervous system. So they are kind of the opposite side of the coin of those oligodendrocytes. The oligodendrocytes are forming those myelin sheaths in the central nervous system, while the Schwann cells are doing that in our peripheral nervous system. I gave you just same thing, more pictures here. So these are all of these cell types we just talked about. Here's kind of illustrations of what they are going to look at like for you under a microscope. And then again, their purpose within the nervous system. So let's talk a little bit about neurons again. The neuron is going to have a few different locations that are really important on that cell. So first you're going to have the soma. That's the cell body. It's going to contain the nucleus and our other really important organelles. We also have dendrites. These are going to receive signals. These are going to have a short little like branching like tree-like shape. You can see these kind of up here around the edges. We also have axons. The purpose of our axon, you can see our axon going all the way down here. The purpose of the axon is to send those signals. We have axon terminals. These are going to be the ends of the axon. This is where we are going to keep our neurotransmitters, which are going to be used as messengers for that cell-to-cell communication. You can see wrapping around your axon, you're going to have these myelin sheaths. The purpose of this is to insulate the axon, and it also helps to speed up the signal. And we'll talk a little bit more about why that speeds up the signal. We also have nodes of Ranvier. These are the gaps between these myelin sheaths. So this is a very simplified view, but it works for us. This is how nerve impulses travel. So essentially your dendrites are going to receive that signal initially. The signal is then going to pass down the axon, and then it's going to cross to the dendrites of a neighboring neuron. So they just kind of hop along neuron to neuron until they get that signal to its destination point. So these myelin sheaths are, it's like a fatty, like a lipid-like substance that is going to be produced by those neuroglial cells, those helper cells we talked about. The purpose of this is to coat that axon of most neurons, not all neurons, but the vast majority of them. And it speeds up the signal. So whenever you have a neuron that has a myelin sheath, it is able to transmit a signal at about 330 miles per hour. Whenever that myelin sheath is not intact, the signal travels about on average 10 to 11 miles per hour. So it is a drastic difference that sheath makes. Let's talk a little bit more about functions of neurons. So we already talked about, we have our sensory or our afferent neurons. This is going to carry the information to the central nervous system via our senses, right? We have interneurons. These are going to interpret the signals from the sensory neurons and relay that information to our motor neurons. And then again, we have our motor or our efferent neurons, and these are going to carry the impulses away from the central nervous system to cause a response. I know we've talked about this a couple times now. That probably means it's important. I would definitely know this information for a test if I was taking this class. Let's talk a little bit about the types of sensory receptors. So sensory receptors are going to be located at the dendrites of the sensory neurons. These are going to pick up sensory information and carry it back to the CNS. We're going to have a few different types of sensory receptors. We are going to have mechanoreceptors. These are going to let us know that there's some kind of physical stimuli. So these are going to be used for like pressure, touch, stress, stretch, motion, and sometimes sound. We also have thermoreceptors. These are going to detect heat and cold. We have chemoreceptors. These detect chemical concentrations. These oftentimes enable like our taste and our smell. We have photoreceptors. The purpose of photoreceptors is to detect light. And then we have nociceptors. And nociceptors, they respond to pain, right? This is how we feel pain, how we feel excess heat, pressure, chemicals, anything that hurts is the responsibility of these sensory receptors. So now that we know the basics about neurons, let's take a look at how they actually communicate with other cells. Okay, so recall that neurons are going to receive those messages at their dendrites, then send the messages at their axon terminals. That's important foundational information to know for this next part to make sense. Neuron communication is just one way. Signals do not go backwards in a neuron, they just move in that forward pathway. So synapses are going to be how our neurons communicate with each other. Neuron communication is going to be electrochemical. That means it's going to involve both electrical charges and chemical signals. So the way this works is we have a synapse or a synaptic cleft. This is going to be the junction or connection between neurons and other cells where they can communicate a message. So synapses can be neuron to neuron or neuron to another cell type. And we talked a little bit about this last week when we were talking about like the neuromuscular junction. So this should be a little bit of a review. So in order for neurons to send a signal, they have to generate something known as an action potential. Do you guys remember this from last week? An action potential is going to be a rapid change in an electrical voltage of a neuron, which is going to start in the soma and travel all the way down the axon of a neuron. The action potential, sorry, I got tongue-tied there. The action potential is going to be generated by the diffusion of charged ions, usually sodium and potassium, across the membrane of the axon. We refer to this as depolarization. If we break that word down, polar, we know that that is creating an area of positivity and an area of negativity, right? We have opposite charges on opposite sides. And if with the prefix D, whenever we are talking about science or medicine, means not. So we are unpolarizing that cell, right? And later on we will talk about repolarization. So we're just taking a polar cell and messing with the polarity of it essentially. Or not a cell, but a polar area and changing that. So as these ions are going to diffuse across the membrane, the charge signal is going to move down that axon. So that charge is just moving and moving and moving across the axon. So let's circle back to this. We touched on this a little bit earlier, but it's a little bit important. So those nodes of Ranvier are going to be the gaps in those myelin sheaths that allow for the ions of the sodium potassium to diffuse in and out of the axon. Because the myelin is going to insulate the axon, the signals are going to jump from node to node. We call this saltatory movement. It's like how a bunny rabbit moves, right? They're literally jumping from node to node of this, and again, that is allowing for faster conduction of that signal. So let's look at these steps in this process. And again, we just went over this with muscular, so I'm just going to do a little quick review. So when the action potential reaches the axon terminals, it's going to signal the release of neurotransmitters into that synaptic cleft. These will bind with receptors on the next cell and continue the signal, right, to eventually cause a response. So the first step in this process is the action potential is going to arrive at the axon terminal. Calcium channels are going to open to allow calcium to flow into that presynaptic neuron, and those calcium channels are going to be stimulated by that action potential. The calcium is going to trigger a release of neurotransmitters into that synaptic cleft. When we talked about the neuromuscular junction, you'll remember that we talked about acetylcholine being housed in those little vesicles, and then through exocytosis, those vesicles release the acetylcholine. This is what is happening in this process. It just will be a different neurotransmitter depending on the outcome that we're looking for. So after the release of neurotransmitters into that synaptic cleft, the neurotransmitters are then going to bind to receptors on the next cell. This is going to lead to either a response or a continuation of the signal. This is the same thing I just said. I just again provided you another picture of it so that you have multiple study tools for the same thing. So we're not going to get super deep into all of the specific neurotransmitters for the sake of this class, but it's still really good information to know. So I'm giving it to you here. So neurotransmitters are going to be chemicals that are released from the axon terminals, which stimulate receptors on the next cell, allowing the signal to continue from the previous cell. We're going to have a whole lot of different ones. Here's some of the important ones. You've probably heard of adrenaline before, right? That is going to be our fight or flight neurotransmitter. We also are going to have noradrenaline. This is going to be kind of like our concentration neurotransmitter. We have dopamine, that is for pleasure. Serotonin helps with our mood. GABA is kind of a calming neurotransmitter. Acetylcholine helps with learning, but also with muscle contractions. Glutamate is going to be our memory neurotransmitter, and then we have endorphins, which can cause kind of a state of euphoria. The neurotransmitter effects are going to be based on what receptors they bind to on the receiving cells. So that's important information to remember. I give you another list here. Again, don't feel like you need to get too deep into like memorizing these specific neurotransmitters. That is not something that we're really going to get that deep into in this class. I just think they're really interesting. So I want to provide you with this stuff in case you do want to go a little bit deeper with this information. I put a crash course video in here. All of the crash course videos I feel like are so helpful. I've used them for biology classes, anatomy and phys, chemistry. I mean, even some of my like sociology and my other basics, histories and things. It is a wonderful channel. All the videos are free. They have a free app. If you download their app, then there's like free flashcards and all kinds of stuff on there. So anyway, I included this as another study tool or resource for you. You're not required to watch it, but it definitely will help you to understand this better. So let's talk a little bit about nerves before I wrap up this lecture, or this part of this lecture. So a nerve is going to be a bundle of neurons, neuron axons, which are going to transmit the electrochemical impulses throughout the body. These are going to branch off of the brain and the spinal cord. And here's some of our important ones. We're going to dive a little bit deeper into this. So most of the neuron cell bodies are found in the CNS, which is going to be in the brain, right, in the brain and spinal cord. The actual nerves of the peripheral nervous system are going to be made up of the axon portion of the neurons. So most of the CNS is going to be the cell body or the somas, whereas the PNS are going to be made up more of the axon portions of the neurons. Some of these neurons can be huge. They can extend over 1 meter in length. Some of them are smaller. There is a lot of variety and diversity with neurons. If you look at the microstructure of a nerve, you'll see that it very much will remind you of muscle tissue. So you're going to have these little nerves. You're going to have coverings around each one and layers and layers and layers. So use your same naming rules when you're looking at these. Like you have your epi, you have your fascicles, you have your peri, you have your endo, right? Everything is the same here that we see in the muscle fiber. So just keep that in mind as you're studying this so that you're not being redundant and studying a bunch of stuff that you already know. Just remember that it can apply to this system just like it does the other one. So again, we've already talked about this. A few times we're going to have a few different types of nerves. We're going to have sensory nerves. These are going to be made up of sensory neurons that are going to bring sensory information back to the CNS. And we're going to have motor nerves. These are going to be nerves made up of motor neurons that are going to bring impulses from the central nervous system back to the body. We can also have mixed nerves. These are going to be nerves that are made up of a mixture of both sensory and motor neurons. So we're going to have a few main types of nerves. We will have spinal nerves. These are going to be nerves originating from the spinal cord. We will have 31 pairs of these. They're going to be numbered by the vertebrae that they extend from. We also have cranial nerves. These are going to originate from the brain. We will have 12 pairs of them, and they are numbered in Roman numerals. I would suggest knowing your cranial nerves. This is not just important for this class. It will come up again, especially if you're doing any kind of health career. Here is a picture of these on an actual brain. I also have illustrations of these to make it a little easier to understand. And this is the Once One Takes the Anatomy final, Very Good Vacations Are Heavenly. That is a mnemonic that a lot of people use to remember the order of these different cranial nerves. So that's why I put this in here for you. So let's talk about a little bit about these different cranial nerves. So we have the olfactory nerve. This is going to be purely sensory. It's going to carry impulses for the sense of smell. We have the optic nerve. This is also purely sensory. It's going to carry impulses for vision. We have the oculomotor nerve. This supplies motor fibers to 4 of the 6 muscles that direct the eyeball to the eyelid and to the internal eye muscles that are going to control the lens shape and pupil size. The 4th nerve is the trochlear nerve. It supplies motor fibers for 1 of the external eye muscles. We have the trigeminal nerves. This is going to conduct sensory impulses from the skin of the face and the mucosa of the nose and mouth. It's also going to contain motor fibers that activate the chewing muscles. We have the abducens. This is going to supply the motor fibers to the lateral rectus muscle. This is going to roll the eyes laterally. So you'll notice a whole lot of these nerves deal with controlling of the eyes. We have the facial muscle. This is going to activate the muscles of facial expression and the lacrimal and salivary glands. This is going to carry sensory impulses from the taste buds of the anterior tongue. The vestibular, sorry, I keep getting tongue-tied on these. I can say it in my head, but it's hard to say it out loud. The vestibular cochlear nerve is purely sensory. The vestibular branch is going to transmit impulses for the sense of balance, and the cochlear branch is going to transmit impulses for the sense of hearing. The glossopharyngeal nerve is going to supply motor fibers to the throat that promote swallowing and saliva production. It's going to carry sensory impulses from the taste buds of the posterior tongue and from pressure receptors of the carotid artery. Our vagus nerve is going to carry sensory impulses and motor impulses to the pharynx and larynx and the abdominal and thoracic viscera. Most motor fibers are going to be parasympathetic fibers that are going to promote digestive activity and help regulate heart activity. Our accessory nerve is going to be mostly motor fibers that activates the sternocleidomastoid and trapezius muscles. And our hypoglossal nerve, these are going to be motor fibers that are going to control tongue movements and sensory fibers that carry impulses from the tongue. So I put those tables in there for you all. Hopefully you find that helpful. You don't have to get too caught up in how you actually test for that in a clinical situation. I just want you to know if they are sensory, motor, or both, and what it is that they do. So let's talk a little bit about the reflex arc, and then that will be it for the first part of this lecture. So a reflex is going to be rapid, predictable, and involuntary. Involuntary there is very important. Response to a stimulus. This is going to occur over pathways called reflex arcs, which are controlled by the spinal cord. A reflex arc is going to be a direct route from a sensory neuron to an interneuron to a motor neuron. Here is a look at this. So here's a simple reflex arc, right? You've all been to the doctor and had your patellar reflex checked. So when that hammer hits, the sensory sensation of that hitting goes in, and then immediately you pull your leg back. So in the next lecture, we're going to take a look at the major anatomical structures of the central and peripheral nervous systems. So don't forget, again, I'm trying to break these into smaller chunks. So this is not the whole lecture. This is part 1 of the lecture, so there will be part 2 as well.